Hello, welcome guys. This is KlassPM and myself Mr. Bilal, and in this video we are going to discuss about the cell cycle, how cell division takes place and what is actually cell division, what is the process of the cell division, how parent cells divide into two cells. Before the cell division, you need to understand the content of the cell, what is in the cell, what are those chemicals, what are those molecules which are present in the cell. Cell has a definite plasma membrane, which is selectively permeable membrane. It allows select molecules to cross through it and restrict other molecules. And apart from cell membrane, there is the cytoplasm, and because cell is an aqueous environment surrounded by plasma membrane, so inside the cell is cytoplasm, and then within the cytoplasm in the eukaryotic cell. I am talking about eukaryotic cell. Apart from cytoplasm, there are cellular organelles such as there is nucleus, there is endoplasmic reticulum, there is Golgi apparatus, there is other lysosomes, ribosomes, etc. There are other organelles as well. And within the nucleus is present the genetic material, and in case of eukaryotes, the genetic material is the DNA double strand. Before explaining the cell cycle or cell division, it is important to mention about the DNA. What is DNA? How it acts as a genetic material, how it carries information from parent to the offspring. Let me precise it. DNA is the DNA stands for deoxyribonucleic acid. Deoxy means it on the second prime position, it does not have oxygen. Simply, deoxyribonucleic acid. There is a lapse of oxygen and the second prime position of the deoxyribose sugar. DNA is made up of three parts. DNA is made up of bases, and bases may be purine or pyrimidine. And DNA, apart from bases, DNA has a phosphate group and sugar as well, and sugar is the deoxyribose sugar. If it is ribose sugar, then you are talking about RNA, and if it is deoxyribose sugar, then you are talking about DNA. Third component is the phosphate group. Phosphate group provides overall a negative charge to the DNA, and overall DNA is negatively charged. And DNA acts as a genetic material has been proved in 1962 somewhere, when it was discovered by Watson and Crick. In their model, they got a Nobel Prize in Physiology of Medicine in 1960 to 62 somewhere. And this DNA acts as a genetic material. It carries the information and it transfers the information from parent to the offspring. And this DNA is located in the nucleus of a eukaryotic cell. And before a cell divides, there is the replication of this DNA before the cell divides. Suppose this is the DNA. Before the cell divides, this DNA has to replicate itself. It means it duplicates itself before being divided, before the cell divides into two daughter cells. And it means that when the cell divides, each and every daughter cell receives exact copies of the DNA. Hence, this DNA is transferred from mother cell to the daughter cell after it replicates itself. Cell division is of actually two types. One is the mitosis, and second is the meiosis. And whatever it is, if it is mitosis or meiosis, cell division actually occurs in two phases. One phase is the interphase, which is the longest phase of the cell cycle, and next phase is the M phase. It is the shortest phase of the cell cycle. If we talk of the interphase head is the subphases like G1 phase, S phase, G2 phase, and M phase has its own subphases like prophase, metaphase, anaphase, and telophase. We will go chronologically what happens in G1 phase, what changes happen in S phase, what changes happen in occurs in G2 phase, and what changes occur in the M phase as itself. So firstly, we will go chronologically. Cell division is of two types. One is mitosis, one is meiosis. Mitosis is also called equational division. It means that whatever amount of DNA is present in the parent cell, same amount of DNA would be transferred to the daughter cells when this parent cell divides. It means that the DNA is replicated and equally separated into the daughter cells. On the other hand, in the meiosis, the overall number of the chromosome is half. There is a 2n number of the chromosome. After the division, the daughter cells receive only half copy of the DNA. So it is a reductional division. In case of mitosis, the different phases are G1 phase, there is S phase, there is U2 phase, then M phase, and M phase has its own phases. In the G1 phase, the cell Before division, it prepares itself for the duplication. It prepares itself for the duplication of the genetic material. Cell has to prepare itself. The replication needs various enzymes, various proteins to happen, and the cell prepares these chemicals, these enzymes in the G1 phase so that DNA can replicate itself very easily. This is cell division. Cell division is mitosis or meiosis. So this is equational division and this is a reductional division. Reductional division means overall number of the chromosome is half when this cell divides in the meiosis. When the cell divides in the mitosis, each and every daughter cell receives exact copy of genetic material from the parent cell. So equal division of the genetic material. So this mitosis is a further following. It has a following subphase. One is G1 phase and one is the M phase. It is interphase and M phase. This interphase is further divided into G1 phase, that is S phase, that is G2 phase, and this M phase is further divided into prophase, then metaphase, then anaphase, then telophase. See, this is the representation of in case of mitosis, and in case of meiosis similar things happen, but their prophase is very long. Prophase consists of its own phases, that is liptoten, zygotene, pachytene and diploten. Otherwise there is no difference. Apart from that, segregation occurs. It is a reductional division. The overall chromosome number becomes half. And this G1 phase, before the cell divides, in the G1 phase, it prepares itself for the duplication of the chromosome. The cell has prepared itself for the duplication. The cell moves from G1 phase to S phase. S means synthetic phase. Synthesis means the synthesis of the DNA. That is the duplication of the DNA. The cell duplicates its DNA. It replicates its DNA. So it replicates in the geometric ratio so that the overall number of the DNA number is increased. The copies are increased. And when the cell has completely duplicated its genome, its DNA, it moves into the G2 phase. G2 means gap phase. And in the gap phase, the cell checks itself whether the replication has taken place correctly or not. If the cell has correctly duplicated its DNA, it has completely duplicated its DNA in a correct manner, then the cell is allowed to move into the M phase. If there is some problem has occurred during the duplication of the DNA, cell checks it in the G2 phase. If there is alteration in the DNA, if there has change occurred in the DNA, then the cell is halted in the G2 phase. It is not allowed to proceed into the M phase, because if it is allowed into the M phase in such a condition, it may turn out into a cancerous cell. It may divide indefinitely, and this may cause the cancer. So when the genome has been completely divided in a correct manner, the cell is allowed to proceed to the prophase. What happens in prophase? The DNA starts to condense itself. It starts to coil itself. So if you see the chromosome is highly condensed form of DNA. It is actually DNA which is highly condensed form. This condensation starts in the prophase. And when the chromosome is fully condensed, the cell is allowed to move in the metaphase. And in the metaphase, you will see a chromosome, fully coiled chromosome you can see in the metaphase. And in the metaphase, all the chromosomes are aligned into the center. There are 22 chromosomes, 22 autosomes plus one sex chromosome. So all chromosomes are aligned into the center to form a plate. This plate is called the metaphase plate. And on the either side of the cell, like this, suppose all the chromosomes are aligned into the center. This spindle assembly, this spindle fiber is they attach the chromosome into the centromere, and in the anaphase, this centriole moves antipolewards and they literally stretch these chromosomes. These chromosomes are broken into the center point. Please see here, it is broken here into the center, and one of the chromatid moves from this side and one of the chromatid moves to the opposite side. Finally, the cytokinesis takes place. The cytoplasm divides, and this plasma membrane is broken, and one cell changes into two daughter cells, and each and every cell receives the exact copy number of the chromatids. And this is the way how mitosis occurs. And meiosis is a bit different. In the meiosis, there is a reductional division. Overall, the chromatids become half in the daughter cells, and you will see in the diagram it is simple like mitosis. but overall the number of the chromosomes are being reduced to half. This is all about cell cycle and cell division. What is cell division? Cell division is the controlled division of the cells, parent cell into the two daughter cells. Because we are multicellular organisms, there is a need of the cell division, because every day millions of the cells, thousands of the cells are undergo the process of apoptosis. Apoptosis means process of cell death. Every day cell dies and those cells has to be replaced. When parent cell divides into two daughter cells, those two daughter cells will replace that cell which is being which undergoes the process of natural death. So this is very important process. So cell division occurs continuously, because continuously in the body some cells are dying on daily basis and those are being replaced by the new cells. And this is all about the cell cycle. Thank you very much.